Okay, everybody, let's make a start. Now, for our last two lectures for this course and my last two lectures in the whole semester, we're going to leave bacteria and think about viruses. Now, before I even do the plan for these two lectures, one thing to bear in mind is that viruses are everywhere. Okay, human beings are probably most interested in animal viruses that cause human disease. But if you look, you can find viruses that infect plants, viruses that infect bacteria, viruses that infect insects. There is no form of life where people have looked that does not have viruses. So they're an important part of the biosphere, and they have probably been around ever since the beginning of life. So what we're going to do for today and Thursday morning, the first lecture will just be kind of general introduction to virology. Really, one of the things that is very important is to be clear about what is a virus, what makes viruses distinct from all forms of cellular life. And never, ever get mixed up between bacteria and viruses, please. We'll have a little look about some techniques in virology. How do we study viruses? Then basic elements of virus structure and the viral replication cycle. Depending on how we go, we might start on the lytic cycle because that kind of follows on straight away. So we might look at the lytic cycle in T4 phage. So in bacteriophages, we'll be looking at lytic and lysogenic replication cycles and thinking a little bit about how bacteriophage can impact on human health. And the end of the lecture on Thursday, we'll be having a look at viral infections in humans with two specific examples, influenza and Ebola virus infection. Okay, so first thing, what is a virus? So when viruses were discovered at the end of the 19th century by Louis Pasteur maybe and others, the only thing that was known about them was that they were infectious agents capable of causing disease. So they were transmissible disease-causing agents that could not grow in culture. Okay, so for all of Louis Pasteur and Robert Koch's work, they were able to isolate bacteria that could be spread out on agar plates. They could get isolated colonies. They could see the bacteria in a microscope. They could culture them and have an idea what they were. Viruses, they were too small to be seen, and they could not be cultured in LB medium or any other type of artificial medium. So that was the first definition of what is a virus, a non-cultivable, transmissible agent. That's kind of a negative definition. The next thing that happened was at the beginning of the 20th century, end of the 19th century, there was the discovery that you could filter an extract containing an infectious virus, and the virus would pass through the filter. So this is telling you that the infectious particle is very, very small. So this became the next classical definition of a virus. It's an infectious filterable agent. So that was a definition about 100 years ago. And during the 1930s, 1940s, beginning of the 1950s, there were a fair amount of experiments on bacteriophage, viruses that infect bacteria, to try and figure out how viruses replicate. And also there was this invention of the electron microscope. So for the first time it was possible to visualize viruses. So new information became available, and this is what led to André Lwoff's proposition of the definition of a virus in 1957. So this is kind of a list of characteristics of viruses that Lwoff proposed that brought together everything that was known about viruses at that time. Okay, so to begin with, he he gave a definition based on the size of the infectious particle. So for Lwoff, viruses are small particles with a diameter inferior to 250 nanometers. That's why they're filterable. They pass through the filter. Their mode of life is, or mode of replication, is that they are obligatory intracellular parasites. They are parasites because they don't possess any of the machinery that is required to generate metabolic energy or proteins. So they are entirely dependent on the host cell to provide these functions. Another thing that Lwoff noticed was that whenever virus particles were isolated, they were composed at a minimum of a protein and nucleic acid, but they only contained one type of nucleic acid, RNA or DNA, but not both. And another thing that had been noticed was that viruses don't replicate by binary division. And one thing that we can add that is not part of the Lwoff if anybody knows why this is happening, if you could send me an email and explain it to me, I would be very happy to find that out. Because when I get to the end of my animations, it makes them all disappear at the end for some weird unknown reason. Okay, another thing that's not part of the Lwoff list, but actually is true for all viruses, is that none of them code for ribosomal RNA in their genome. So that explains, or it's related to the dependence of of all viruses on the host for protein synthesis. And this is really quite an amazing observation because our ribosomal RNA, this is one of the most conserved molecules in all forms of life. Okay, you can use it to establish a universal phylogenetic tree for every living organism. And no virus has this. So this is some microbiologists who are interested in fundamental questions in evolution, like Patrick Forter at the Pasteur Institute. They even said that this is, this is the first fundamental bifurcation in the history of life between replicators that have ribosomes, that's cellular life, and replicators that don't have ribosomes, and those are viruses. And ever since then, we've got, these two forms of nucleic acid replicators that are competing with each other. Okay, so what I'm going to do now is take a look at each of these characteristics one after the other. And this will be the last one because it's going to oblige us to think about how viruses replicate. So that will come later. But to begin with, we're going to look at these criteria because that's what was known in 1957, and since then we've got new observations. So some of these criteria proposed by Lwoff are not always true for all viruses and not necessarily sufficient to distinguish viruses from bacteria. So looking at the criterion of size of the infectious particle. So the smallest virus particles have a size of maybe about 20 nanometers diameter. Most of them will be in the range of, 50 to 150 nanometers diameter. And there are some large viruses like pox viruses. This is about, 200, this bar here is 200 nanometers. So it's just under 250 nanometers in diameter, 300 nanometers long. Giant viruses have since been discovered. However, if you just look at electron micrographs here and here, there's not much to distinguish large complex viruses like pox viruses here from this type of biological object. So this is 100 nanometers. They're shown to the same scale. This particle is about 150 nanometers in diameter, maybe 500 or 600 nanometers long. And in fact, it's a bacterial cell, or a mycoplasma, one of the bacteria that do not have peptidoglycan cell walls. And just looking at the size and the complexity of the particle, not really enough to distinguish the largest viruses from the smallest bacteria. There's a little bit of overlap at the extremes. So some virus particles are bigger than this. That's the message here. Obligatory intracellular parasites. Yes, all viruses are, but that's also true for some bacteria. So Mycobacterium leprae, Chlamydia and Rickettsia, all can only grow inside a host cell. They can't grow in a culture medium. So once again, this definition does correctly describe all viruses, but it's not sufficient on its own to distinguish viruses from all bacteria. And no enzymes for the synthesis of proteins or to generate metabolic energy. Especially true for protein synthesis. No ribosomal RNA, no ribosomes inside viruses. Every form of cellular life has ribosomes, even dormant forms like bacterial endospores. They have some ribosomes there. So that's a big difference. Okay, most bacteria have everything they need. They generate at least their own metabolic energy. A little bit of an exception with Chlamydia because they exchange ADP, they export ADP, and they import ATP from the host cell. So they don't really generate metabolic energy themselves. All viruses require the host to produce metabolic energy and proteins. And the nucleic acids, you know, this goes back to the fundamental functions in a living cell. So in cells, the genome is in double-stranded DNA. And in order to express the genome, every type of cell needs messenger RNA, transfer RNA, and ribosomal RNA. Bacteria, eukaryotes, yeast, and once again, even dormant forms of cells, they all have these different forms of nucleic acids. However, most virus particles, let's say almost all of them, only have one type of nucleic acid. Either double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA, but not both. And this is just to show you electron micrographs of different types of virus particles. And at least for icosahedral naked virus particles, you can't really tell by looking at the outside what the nucleic acid is going to be on the inside of the particle. So what is the message here? Okay, once again, it's just repeating, okay, viruses don't make their own proteins, so they don't have to carry any of these things. And they don't necessarily replicate their genomes in the same way as the host. So genomic nucleic acid is much more varied than in the host cell. Okay, so those are the kind of theoretical characteristics of viruses. So how do we study them? The big problems are that they are too small to be directly visualized. You can see them only by electron microscopy. And they only grow inside other organisms. So the first thing you need is a system for production. So this will be some other kind of host organism. And then if you can purify them, then you can have a look and see what they're made of. visualize them by electron microscopy, and analyze the components of the virus particle. That's one way of studying viruses. Another thing that can be done is to analyze them in situ, that is, infect an organism, infect a cell, and then have a look and try and detect viral proteins and viral nucleic acids inside the infected host. So for that, you'll use immunochemistry, specific antibodies to detect viral proteins, and molecular biology techniques to detect virus nucleic acids. Okay, so for production systems, okay, the first ones that were used were whole organisms. Pasteur in the 1880s was able to cultivate rabies virus by making an extract from the brain tissue of infected animals and injecting it directly into the brain of another animal. So he was able to passage the virus in rabbits, I think, not actually in mice, and produce an attenuated and inactivated virus after multiple passages in a live animal. That's the same way that plant viruses were discovered. So initially, to propagate plant viruses, you would get an infected leaf that had symptoms, grind it up, and then just scrape the leaf of another plant and transfer the virus to another whole organism. So these type of production systems are still useful if you want to look at models of infection and find out how viruses cause disease and cause pathology. But they are not so good for understanding the fundamental processes of virus replication. For this, you need to have a simpler system. You need to have the cells that are isolated, the virus that's isolated, put them together and see what happens. So in order for that to happen, you need cell culture. Now, of course, the first type of cell culture that was available were cultures of bacteria. This meant that bacteriophage, viruses that infect bacteria, were the easiest ones to study in order to get information about virus replication. So a lot of the fundamental work that helps us understand how viruses replicate were done first in bacteriophage. And then from the 1940s onwards, it was possible to culture mammalian cells in immortalized. So these were the first immortalized cell lines. First one was looking for the name, it was a mouse cell line was the first one, and then the first immortalized human cell line was, anybody know? HeLa cells, isolated from a woman with very, very severe form of cervical cancer. And that was the first immortalized human cell line. It was very important for virology because that meant that there were human cells in culture that would survive forever, and they could be used to grow viruses. And this was what was essential for cultivating poliovirus and producing the vaccine. Now, whatever your production system, okay, you inoculate the virus, it will replicate, you'll have a lot of copies of virus afterwards. So then you'll be able to harvest the infected animal, the infected plant, or the infected cells, grind them up, make a raw extract. The next step is to purify the virus from the raw extract. And the classical system for doing that is to use ultracentrifugation on a density gradient. So you start out with a density gradient of sucrose, for example. So your density is highest in the bottom of the tube and gets lighter as you go up. And you put in there a mixture where you'll have soluble proteins, large cell fragments, like bits of cell organelles that are mashed up, and somewhere, and you'll have virus particles all mixed up. You set up your tube, you put it in the ultracentrifuge, spin it round at, I don't know, 130,000 G for 24 hours, and the different components in the mix will be pulled down the gradient and form bands according to their sedimentation coefficient and their density. So all these large fragments from the cell are going to go down in the pellet. Soluble proteins, they might enter the gradient a little bit, but they will stay high up in the gradient. And at some point, virus particles will concentrate at one particular point in the gradient and form a band. You can then harvest. And if you have a lot of material, this is what you'll get at the end of the experiment. You've got pelleted material out here, and here you can see a band of material somewhere in the gradient. You can harvest this band and analyze it by electron microscopy to see what the particles look like. So this is an example. This is pox virus particles. And then you can analyze these particles by any number of biochemical techniques. So SDS phage, you can analyze the proteins that are present in these particles. So the example here is a little bit unusual. You can see there are a lot of different bands in this gel. Pox virus particles are very, very complex. So it's a little bit exceptional. What you would often find if you run a band like this on SDS phage, you might find that you have one protein that makes up the virus. Or you might have 2 or 3 viral proteins, or 10, something like that. Most virus particles are made of, 1 to 10 or 2 to 20 different proteins in the particle. And that's how we know. Okay, so you can get viruses to replicate in some kind of host system. You can purify them. You can visualize them by electron microscopy and analyze what they're made of by biochemical techniques. So what does that tell us? Okay, it tells us the fundamentals of virus structure. So some viruses are called naked or non-enveloped. So they're composed just of nucleic acid in the middle and a protein capsid around the nucleic acid. And others are enveloped. So they have a nucleocapsid in the middle, nucleic acid, protein capsid, and then a lipid membrane called the envelope, which is derived from one of the host cell membranes. Now, among naked virus particles, there are 2 different types of morphology. Particles that are kind of stick-shaped, which have a helical symmetry, and particles which are roughly spherical, which have icosahedral symmetry. And then in bacteriophage, you have these 2 elements that are put together to form tailed phages. Within the envelope viruses, kind of easy way to classify these is by calling them simple or complex. So with a simple virus particle, you've got one of these 2 types of nucleocapsid on the inside, and then you've just got an envelope on the outside. So here you've got a nucleocapsid with helical symmetry and then an envelope, and here you've got an icosahedric nucleocapsid and then an envelope on the outside. And complex particles, well, that just means that inside the particle, it doesn't really conform to either one of these 2 simple models. Okay, so those are the basic structural elements of viruses: genome, nucleic acid, capsid, and the envelope. So what are their functions and how are they formed? Now, the basic function of the capsid is to protect the genome. Okay? All viruses need to replicate. At some point, they're going to be released outside of the host cell, and they will have to infect another cell. Just a naked molecule of nucleic acid is not going to last very long. In your body, you've got lots of DNase 1 out in serum. You have RNAs in extracellular fluid. And free nucleic acids do not have a very long half-life. So the basic function of the capsid is to protect the nucleic acid from degradation, either chemical or biochemical. So the whole point is to try and cover up the nucleic acid and protect it. And really the idea with helical capsids is that, okay, we've got a long linear molecule, DNA or RNA, nucleic acid. It's a long linear molecule. What I can do to cover up and protect this nucleic acid is just get a protein that's going to form a spiral or helix around the nucleic acid. So if I do this, I'm going to end up with an elongated particle, and I can protect all of the nucleic acid. So the big classical example of this is that is, for example, tobacco mosaic virus, which you can see here. So tobacco mosaic virus is composed of an RNA genome, and then I don't know exactly how many, but several thousand copies of one capsid protein. So on its own, the capsid protein forms a double disc. When it interacts with the nucleic acid, these two discs are going to be décalés, shifted, one with respect to the other. So you end up with a free face here, and another subunit of the capsid protein is going to come and stick here, but there'll still be another free face. So another one's going to be added on, then another one, then another one, then another one. So it just carries on until there's no nucleic acid left. You get this kind of particle. So that works. It's a very simple system, and viruses can, successfully encapsulate all of their nucleic acid. So you find this very often in plant viruses. There's a catch. Okay. If, as a virus, you want to become more complex and have a genome that has more functions, your genome is going to get longer and longer. Now, if you have a capsid that's got this type of morphology, that means that the bigger the genome is, the longer the particle is going to have to be. And that is going to end up causing a problem. Okay, so even here, you've got something that's about, I don't know, 20 or maybe 15 nanometers in diameter, and it's already 200 or 300 nanometers long. If you carry on increasing the genome size, the particle is going to become longer and longer and longer. And in the end, it's going to become physically unstable. And it's just going to break in half. So viruses that have got this type of structure have a kind of limit on the genome size that they can have, because physically the particle will become unstable. That's one problem. So is there another solution? Well, yes, there is. So in long helical capsids with helical symmetry, okay, the nucleic acid, you consider it to be a line in 2 dimensions. But of course, a linear molecule can be folded up in 3 dimensions and wound up like a ball of wool or something, okay? So in that case, if you wind up the DNA in 3 dimensions, or RNA, then you can try and, so this will be kind of spheric, you get a ball kind of shape. And you want, in that case, the capsid is going to go around it. So this is the kind of idea, or the objective, or the concept behind capsid with icosahedral symmetry. So here the capsid proteins create a kind of hollow sphere, and the genome goes inside. Now the problem here is for a limited number of proteins to form a spherical shape. Now the simplest system is if you've just got one virus protein. 3 copies can join together to make a kind of triangle, and 20 copies of the triangle make up an icosahedron. That's why it's called icosahedral symmetry. So that's the simplest viruses with icosahedral symmetry have this kind of structure. 60 identical subunits, just one virus protein. And if you want to make the particle bigger, you've got to put a few more different proteins in there. So the bigger they get, the more complicated the structure can be. Now what you can see here is that this is from the picornavirus, poliomyelitis virus, or something like that. There are 3 virus proteins, okay, in green, red, and blue. And you can see perhaps that the blue proteins, this is VP1, form a kind of pentamer at the vortex here of the icosahedron. And in the middle of one of these triangular faces, VP2 and VP3 form a hexagon. So a lot of icosahedral viruses are formed with one protein makes a penton, and then other proteins make a hexon. Okay, so those are the basic functions of the capsid. Protect the nucleic acid. Now in envelope viruses, there's another basic function of the capsid proteins, which is to interact with the host cell. So here's your host cell. It's going to have a receptor, and the capsid proteins will bind specifically to the host cell in non-enveloped naked viruses. Now in envelope viruses, of course, the nucleocapsid is on the inside. So it can't interact with anything on the outside of the virus anymore. It can't interact with the host cell because it's got the lipid membrane on the outside. So that means that viruses have got to have another protein. And those are the envelope glycoproteins. So in an envelope virus, the strict minimum that they must have is nucleic acid in the middle, a nucleocapsid protein, and at least one envelope glycoprotein in order to interact with the host cell. So that's the function of the envelope glycoproteins. Now many viruses, envelope viruses, also have some kind of protein that is in between the nucleocapsid and the envelope, and we call these matrix or tegument proteins. Their function is often to help just with the physical integrity. Okay, they keep the envelope attached to the nucleocapsid. Okay, so this is the summary for up to now. You have to have a clear idea of what the definition is of a virus in these kind of terms and in structural terms as well. Okay, what is a virus made of? Nucleic acid, capsid, maybe an envelope, possibly an envelope. And should have some kind of idea of how we study viruses. Okay, so that's been half an hour already, so have a little think about these questions. See what you think. Okay, ready for all of these ones? So, no? Yes. Okay. So all envelope viruses have at least one envelope glycoprotein. Who says yes? Who says no way? Yeah, it's true, at least one. Virus capsids are composed either of polysaccharide or polypeptide. Who says yes? Who says no? No, okay. This is because capsid, capsule for bacteria, people get mixed up sometimes because the words are very similar. Okay, capsid, this is in viruses, it's always protein. Capsule is bacteria, it's on the outside, and that's true. Polysaccharide or polypeptide is a bacterial capsule, so don't get mixed up. The main function of the capsid is to protect the virus genome. True? Yeah, it's true. 4. Okay, now these ones are all quite simple. These two, you might need to think about them a little bit. Naked viruses are more resistant to detergents than envelope viruses. Who says true? Who says false? Slight majority for false. Who says, oh, we can't know this? You can know this. You have to use your knowledge of biochemistry. Okay, people who said true. Why? Why is this true? You just a guess. Yeah. Maybe. Yeah. Okay. I understand your thinking, but it's not the. Yeah. Yes. The envelope is made out of. It's a lipid membrane, right? Detergents disrupt the lipid membrane. True for cells. It's also true for envelope viruses. So think about basic biochemistry here. Okay, that's definitely true. Okay, so naked non-envelope viruses are more resistant, especially to detergents. So, for example, it's important for control measures. Simple hand washing with soap will be enough to inactivate envelope viruses like influenza. It probably won't be enough to inactivate a non-envelope virus like hepatitis A virus, which is foodborne, can give you hepatitis. Finally, okay, viruses are not living organisms. Who says true? Who says false? Okay, so most people think viruses are not alive. Probably, I guess most microbiologists agree. They're it's weird, it's part of the biosphere, but they're not alive. So okay, they're not alive, but are they inanimate? Okay, is it, you could say, if you say it's not alive, is it in the same category as a crystal of salt or rock or something? I don't think so. So it's kind of in between. It's not really alive, but not really inanimate either. So anyway, no correct answer for that one, but anyway, they occupy an interesting place, I think, in biology. Okay, yeah. Hang on. Let me go back to this actually, because I've just let's go back to this one a little bit. Okay. Okay. Imagine this is a host cell, and here's the host cell genome. Now, there are some viruses, especially bacteriophage, that can kind of switch. They can be maintained in the host cell as a plasmid form or provirus form integrated into the genome. So you might like to put viruses in a different category and say they're not alive. But you, as we'll see, yeah, maybe in a while, but later on, there's a kind of continuum between viral genomes and host cell genomes, especially in bacteria. So this bacterium is alive, right? It's a living cell, and it depends on its genome. But part of this genome will be derived from viruses. So there's a kind of, at least a genetic continuum between some virus genomes and some host cell genomes. So difficult to say, you know, are they alive, are they not alive? On the other hand, you wouldn't call a plasmid alive, would you? So I suppose you'd put viruses in the same category. Okay, so thinking about virus replication now, one of the criteria of Lwoff was that viruses don't grow by binary division. And what that means is that this is an important thing because it's a fundamental difference between viruses and cells. So all cellular organisms, at some point, you know, you've got one cell, it grows a bit bigger, and then it will divide into two daughter cells. So this is true. It's true of bacteria. It's true of cells in our bodies, and there really aren't any exceptions. So you might imagine that if viruses did the same thing, you would start out with a virus particle, it would get a bit bigger, and then eventually these two virus particles would separate and you would have two daughter viruses. Now, this does not happen at all. Okay, so viruses don't grow like this. At some point in their life cycle, they get entirely disassembled, and then later on they reassemble, like, I don't know, a Lego kit or something. Okay. Now, the first indications that this is what was happening came from experiments with bacteriophage. Classical experiment at the end of the 1930s. Ellis and Delbruck. And what they were trying to do was establish the growth curve for viruses, just like, you know, what we did for bacterial growth curve in the practical. So you inoculate some kind of culture with a phage. So for bacteria, if you want to do this, you have to give them a culture medium. But of course, viruses don't grow on culture medium. They can only grow if they have a host cell. So you have to inoculate the phage into culture of bacteria. And then you sample at different times, count the number of phage that are in the culture, and this will give you a growth curve. So here are some of the figures from the original paper in 1939. So this is time, and this is the number of phage on a log scale. And they got this kind of one-step growth curve. Okay, so a lag phase, a liberation phase, and then a plateau. Now, to understand what is going on here, you really have to understand the technique. So this is how to count bacteriophage in plaque forming units. So just like what we did with the bacteria in the practical class, you start off with your sample, make a series of 10-fold dilutions. So you get 10 to the 1 to 10 to the 6-fold dilutions. Then inoculate them into bacteria and soft agar. So on each plate you'll have some agar and then a kind of mix with bacteria and the soft agar on the top, and from time to time a phage. Now what is going to happen is that this cell will become infected, and later on during the overnight culture it will release other phage. Because this is in soft agar, these phage can't diffuse all over the plate. They can only diffuse a short distance. It's like the antibiogram, right? So with the discs, the antibiotics didn't go everywhere. They diffused in a small area. So they can infect the neighboring bacteria. And then these bacteria themselves will be lysed. So when you look down on the plate like this, what you'll see is you'll have bacteria that grow all the way over the plate, except for some zones which will be lysed. And that's what you have here. Okay, so at the end of the experiment, you've got this. If you've got a lot, you'll have the whole plate will be cleared. Then you have too many plaques to count. Here there are 83 apparently. Here there's 8. So you just count them up, multiply by the dilution factor. You've got 8.3 times 10 to the 4. And then you account for the fact you've added 0.1 ml. So here you've got 8.3 times 10 to the 5. So you can count the number of phage in a sample. But it only works, okay, there are a couple of drawbacks with this. Okay, it can only work if these viruses are lytic. Okay. Now, I would like you to think about this in this assay. What does a plaque represent? This kind of hole here in the lawn of bacteria. So another way of thinking about this is, if you have this kind of situation, how many plaques would it give you? So think about options 1, 2, and 3. If you find that it's not obvious, you can ask the people sitting next to you, see what they think. Okay, so has everybody chosen one option between 1, 2, and 3 here? Yeah? No? Okay, ready to let me know what you think? Okay, right, who says 1? If you have a plaque like this, there's 1 infectious phage in the medium, but not an infected bacterium. Who says that? Nobody. 2, 1 infectious phage, whether it's free in the culture medium or inside an infected bacterium. Who says that? One person. Or 3, either one infectious phage in the medium or one infected bacterium. Doesn't matter how many are inside it. Who says that? Maybe about 10 people. So here, how many plaques would you get here in that case? Well, okay, the answer is 3, right? That's true. This is the correct answer. So here you would get how many plaques? What are the options? What are the options for this? Okay, this is free phage in the medium. This is an infected bacterium with 4 on the inside. Okay, so how many plaques would that give you? Anybody got any ideas? One. Anybody say 1? No. Anybody say 2? No. Anybody say 5? Anybody say 8? Nobody. Anybody say 4? Okay. Hang on. This is true. Number 3 here. Okay. So that means each one of these, they will form 1 plaque. So 1, 2, 3, 4. And then you've got 1 infected cell. 5. 5 is the right answer here. Okay. Why doesn't it matter how many are inside the infected cell? Okay, when you set this up, okay, what's going to happen is if you start with an infected cell, well, it releases phage, but only locally, right? So it can't give you any more than 1 plaque because all of these guys, they are exactly in the same place. Okay, so 1 infected cell will give you 1 plaque. And each free phage, 1 plaque. Everybody clear on that? Now I think it's not easy, this idea, but it's important to understand the interpretation of these growth curves. Okay, so this is what Ellis and Delbruck found. Okay, so they would have a latent phase. Okay, this is the time, and this is what they found in their growth curves. You know, things would the titre would not change. It would go very rapidly in the release phase, and then it would stay stable. There's no more bacteria to infect. They've all been killed. Now, the first model that was proposed to explain this is that, okay, we've got the phage. It looks like this. It goes into the infected cell. At the beginning, you've got 1 phage inside the infected cell. Then it's going to grow. It's going to replicate inside the cell. And then these cells will be lysed, and you'll have a very, very rapid release phase. And you get a whole bunch of phage in the supernatant, and that's what you see here. So this was the model, okay? Now, the way to confirm this model, what you would like to do is have a way of counting the phage that are inside the bacterial cell to confirm the model or refute it. And in fact, it's possible to do that by using chloroform, which will lyse the bacteria, but fortunately these bacteriophage are non-enveloped, just nucleic acid genome and a protein capsid, so they resisted chloroform lysis. So then you get rid of the infected bacteria and you liberate the phage that are inside. Then you can count them. And the big surprise here is, okay, the dotted line is what they saw before without chloroform. With chloroform, they got this red line, solid red line. And the big surprise is that at early time points, it's right, 0. There are no infectious phage anymore. And this is what was called the eclipse phase. So what does that mean? That means that just after infection, the phage itself seems to disappear. So there isn't any phage or virus, you just have an infected cell. That's why without chloroform you still get plaques, because you have an infected cell, but you don't actually have any viruses, any phage particles inside it. Then what happens is that phage particles are formed inside the cell, so that's why this curve comes up again after the eclipse phase. You get higher numbers until before lysis. So this was a key experiment showing that shortly after infection, the bacteriophage ceases to exist. This is what shows you that viruses don't grow by binary cell division. Because all the time when you've got cells growing, okay, you start with one cell, you've got two cells afterwards. But at every time point, you at least have a cell somewhere in the culture. Here with viruses, that's not true. Shortly after entry, the virus doesn't exist anymore. So that's the meaning of this experiment, the importance of that result. And it was really the start in understanding the different steps in virus replication. Now, some of them can be visualized by electron microscopy. So these are going to be images not of a bacteriophage, but chlorella viruses that infect eukaryotic cells. So the first step is attachment of the virus onto the surface of the host cell, and then entry either of the virus or just of the virus nucleic acid. Okay, so here you can visualize the steps in attachment and entry of these chlorella viruses, and you can see that at the end of entry, the capsid stays on the outside, and it's only the nucleic acid that goes inside the host cell. That's why you don't have any infectious virus anymore. What do you need to have a virus? You need the nucleic acid and the capsid, both together. But at early time points, these two elements are separated. That's why you don't have any infectious virus anymore. So once you have this separation of capsid and genome, that's called decapsidation, that's when you have the eclipse phase. Can't see any viruses anymore. And later on, you have assembly of viruses inside the infected cell. You can see them here. You get more and more, and then in the end, well, the host cell is lysed, and then you have release of these these new virus particles. So these are the basic steps in the replication cycle of any virus. Okay, it was first worked out in bacteriophage, but it's true of all viruses. All viruses during their lytic cycle, that is. First steps are attachment and entry, then decapsidation. At some point, the virus genome is released from the capsid. After decapsidation, that's when you have the eclipse phase. Two things that must happen is that the genome has to be replicated, and you have to get gene expression inside the host cell. So you get a lot of copies of viral nucleic acids, a lot of copies of viral proteins, and then you put them all together to produce new virus particles, which will then be released. So you start with one virus particle outside the cell, you end up with a whole bunch more. Okay, so to put this, or to bring this to a human scale, I want you to try and imagine the scenario. Okay, me driving my car, I'm a virus. Okay, the genome, that's me, and the capsid is my car. So I'm driving around, and I go to the Chantier de l'Atlantique in Saint-Nazaire, Chantier STX, and I attach. I go up to the entrance. Okay, and I've got some kind of ID card that I show to the security guy on the gate of the shipyards, and he lets me in. That's it. I've attached and I've entered into the shipyard. Once I get in there, I get out of my car. Decapsidation. Then what do? I go into like the central place where they control everything in the shipyard, and I reprogram the whole system to stop them making ships, and they start making cars. and I transform all of the employees of the shipyard into a copy of me. And then all of my clones get into a car, and we all drive out. We blow up the shipyard when we leave. Boom! Okay, so that's a lytic cycle, lytic replication cycle. That's what happens. And we all drive out and we go and infect other factories somewhere else. Okay? So that's the lytic cycle of viral replication. Okay? Right. So for virus replication, what you need to remember now. Okay, this is really a different process, not the same thing at all as cellular division. And the first experiments that helped us understand this were simple experiments just looking at the bacteriophage growth curve. And this, you really should know this, okay? You just have to learn these steps and remember them, please. True for every single virus. Okay. I think what I'm going to do now is skip ahead a few slides because we're on this topic. So I'm going to skip over this and come to here. Well, that's where we've just been, okay? And think about how bacteriophage accomplish the different steps in the replication cycle. Okay, the first step, attachment. Every virus is adapted to a particular host. So it's very important for every virus or for every bacteriophage to infect the right cell. Okay, if you're in the ocean, down at the beach, you're a virus that likes to infect algae, cyanobacteria. There's no point if you bind onto the surface of a piece of seaweed. So you have to bind to the right cell that can support the replication of that particular virus. So, and this occurs because there's always some kind of receptor on the host bacterium, and it's specifically bound by an attachment protein on the phage. Okay, so what are the receptors on the host cell? It's got to be something on the outside surface, okay? So bacteriophage T4, that's this guy, looks like this. At the end of the tail fibers, this is the attachment protein. The receptor on the host bacterium is OMP-C. OMP is outer membrane protein. It's a porin in the outer membrane of the bacterium. Okay, so these guys infect Escherichia coli, so they infect gram-negative bacteria. So remember your structure of gram-negative cell wall, peptidoglycan, plasma membrane, outer membrane, LPS, and sometimes you have porins. So OMP-C is on the outer surface of the bacterium. It's available for binding. Similar thing happens with lambda-phage. Here the J protein is at the end of the filament, the end of the tail filament. Lam-B is the permease for maltose. Once again, in the outer membrane of the bacterium. M13 phage binds onto the conjugation pilus. So, okay, you have a pilus that is here, and M13 is going to bind here. Binding up here is not going to be very good for the next step, which is entry, right? What is an interesting characteristic of the conjugation pilus in bacteria. So I showed you some videos of what it can do, if you remember. It can extrude and it can be retracted. Okay, so type 4 pili can do this. So this is what happens. The binding seems to induce retraction and internalization of the pilus. So when this happens, the phage is pulled into the cell. Okay, so those are examples of bacteriophage that infect gram-negative bacteria. So the receptors are structures that are on the outside. What about this one? This is a phage that infects Bacillus subtilis, gram-positive, and its receptor is this protein UEB. So here's your gram-positive cell wall, and the UB protein is like this. It's a transmembrane protein. It crosses all of the peptidoglycan layer, and it's got part of it is accessible on the outside of the bacterium. Okay. So the host cell receptors in bacteria are something that's on the outside of the bacterium. Okay, outer membrane proteins, F. pilus, or large transmembrane proteins in gram-positive bacteria. This is one of the roles of the capsule in bacteria, because the capsule is out here, and this will prevent bacteriophage from gaining access to these receptors. So having a capsule is one of the bacteria's mechanisms to resist infection by a phage. Okay. Attachment on its own is not enough. Okay. If a virus just attaches to the host cell, it stays on the outside. It needs to get on the inside in order to be able to replicate. So all phage and all viruses have a specific mechanism for entering the cell, or at least getting their DNA inside the cell. So in phage T4, this occurs firstly, okay? First step is, okay, the filament will bind to OMP-C. Then this base plate will penetrate the outer membrane, release phage lysozyme, which would degrade the peptidoglycan layer, and then finally the genome is going to be injected into the cytosol of the bacterium. And I can show you this in a rather more impressive way here, hopefully. Okay, so here's a little video that you can find on YouTube. Now, this is a good video because all of the things that you see are accurate down to the atomic scale, well, molecular scale. Okay, so the first step is binding onto the OMP-C proteins. Here again, tail filaments will deploy, bind onto OMP-C, then you get contraction of the tail, and hydrophobic domains will penetrate the outer membrane, and this releases phage lysozyme, which are these green things here, degrade the peptidoglycan layer and allow the tube to penetrate into through the plasma membrane. So, okay, a lot of work has been done on this. So all of the structures here are accurate. Okay, all of the structures have been determined, and the video is done using real data. Okay, we have 10 minutes more, and I think we could go through the important parts of gene expression and genome replication for T4 phage. Now, at the end of entry, okay, you saw that the capsid stays on the outside, only the virus DNA gets injected. So you have virus DNA that's entered the host cell. Two things need to happen. It's got to replicate its DNA, and it's got to express its genome, produce proteins. And once you have both of these things together, you can produce new phage. So here is what happens. So first thing, you've got DNA that comes in. Very quickly, promoter regions in the phage genome will start to be bound by the bacterial RNA polymerase with the sigma 70 subunit. So these early mRNAs code for DNA polymerase and a DNase. So this nuclease will degrade the host cell DNA. Can anybody see a problem with this? At the same time, you've got a phage. It's expressing an enzyme that's trying to polymerize DNA and an enzyme that's trying to degrade it. How can this work? It's like you're trying to build something and there's somebody just next to you who's pulling it all apart just afterwards. Well, it works because the phage DNA is chemically distinct from the host cell DNA. And instead of cytosine, it uses hydroxymethyl cytosine. So one of the nucleic acid bases is chemically distinct. So at the same time, hydroxymethyl cytosine synthase. Another phage enzyme modifies the nucleic acid bases, and the hydroxymethyl cytosine is used. So you'll get, cytosine going to cytosine, and this is used as a substrate by the host. So bacteriophage does not degrade its own DNA. It will only degrade the host cell DNA. Okay, you've got degrades the host cell DNA. The DNA polymerase will replicate its own DNA. Afterwards, after DNA replication, you'll have late mRNAs made. Now these will be the structural proteins of the virus and also proteins involved in lysis of the host cell, lysozyme and a holin which will penetrate the plasma membrane. Now it's very important that these late messenger RNAs are not expressed immediately, because if you've got like the lysozyme that is expressed straight away, the host cell is going to explode before you even get any new particles made. So control of the expression of these late mRNAs is very important. So these are the early mRNAs or early genes. One of them codes for a phage protein called GP55. This acts as a kind of sigma subunit for the host RNA polymerase, and this is required for the expression of the late messenger RNAs. So without this, the late messengers can't be expressed and you can't lyse the host cell. Okay? So that's the lytic cycle for bacteriophage T4. And what you should know is, okay, what are the different proteins involved? What are their functions? Okay, nothing. Okay, assembly and release, don't really need to say anything about that. So just in a general way, okay, what is interesting about this is that different components are made independently. Okay, so you get the heads that are formed first, then the tails, then they get together, and then finally the fibers are attached. And then once the whole thing has been formed inside the cell, then you get lysis. Okay, it's 5 minutes early, but I think it's about time to stop really. So I'll let you go. So what I'll be doing on Thursday is a little bit more general stuff about bacteriophage and no, yeah. I'll go back and do general things about bacteriophage, then the lysogenic cycle, and then we'll do human viruses. So tonight, I promise this will be the last microbe of the week, and I will not forget to send out the question. So what time should I send it? 7 p.m. or 8 p.m.? 7. Okay, 7 p.m. Okay, so that's it. Bye.